everyone, good morning, and welcome back to another episode of Health Discourse with Sushant and Sakshi. Hi, good morning, Sushant.
Good morning.
So, it's been quite a long time, and we are doing this podcast. I think this is the first podcast of the year. Yeah.
Yeah,
So,
yeah.
would you like to tell the audience where we have been, what we have been doing at Gene Fithletics?
Yeah, so since 2025, there have been a lot of expected and unexpected developments, right? Both good and bad side, right? And again, let's talk about good sides. There's no point of discussing the bad aspects, right? So ideally, we started working on longevity, and I don't know why our actions always precedes the industry's action. So probably when we started longevity, after a couple of months after that, probably and we have seen this longevity and biohacking wave, right? And biological age. Although we started operationalizing this since January, but we have been working on this for last 4 years, right? And it has been hell of a ride in terms of collecting a lot of molecular data, and now we have got into the cellular aspects of health, cellular aspects of aging, how specific biochemical interactions at the body, whether it is microbial, whether it is human host or mitochondrial or oral microbiome, gut microbiome, it is impacting the cellular functions, right? And we could measure a certain pathway which lighten up in response to a lot of environmental and stress related and inflammatory related factors which directly impact the cellular function or the cellular homeostasis, right? So again, that was what we started. Now we could, from our whole body biochemistry solution, we are also giving you your amino acid profile, so we can tell you how your microbiome is interacting with various amino acids, mostly essential and some semi-essential, right? We are now can also giving you a score of cellular senescence, right? And again, which is again one of the hallmark for cellular aging, right? And again, we have revamped our model based on the new data sets. We have further fine-tuned and modified our deep learning model, right? Now we have more and more data sets. So as we speak, we have more than 10 billion of molecular data sets right now, right? And again, more than 500,000 of phenotypic data, metadata from the patients, right? And again, we have been tracking using our technology to, on the real-time basis, how our interventions are impacting various metabolic and immune and other cellular pathways, right? And how it is uh, driving an individual away from a disease, right? So these have been few things what we have done, built. Obviously, we are coming up with this year we should come up with certain early detection diagnostic tests for cardiovascular disease and chronic kidney disease, right? So for people who want to know about biological age, probably we'll be discussing today about biological age, and also demystifying why the other biological aids lacks efficiency and accuracy and so-called comprehensiveness in their assessment of the biological age clock, right? But this is right now available to everyone, the current, past, and future customers at no additional cost, right? So this is a part of our product from with our full body biochemistry, that is Aura Pro, where we measure your oral microbiome functions, gut microbiome functions, your mitochondrial functions, your cellular markers, cellular pathways, as well as now aging, right? So that's what we have been for the last 3 months, right? And yeah, we also invited certain personalities which are experts in their respective fields. So we had this excellent podcast with Professor Thomas Seyfried 2 months back, right? On I think it was end of January, I believe. Yeah, as far as I remember.
It was on cancer that,
Yeah,
cancer
yeah.
is a metabolic
Yeah, so
kind
we
of
discuss we
therapy.
discussed yeah, we sought his views because he had been into the cancer research for so many years, right? And again, that was an insightful discussion on how we are solving cancer in a wrong way, and that's our basic premise and fundamental, right? We look cancer as an outcome of damaged respiration and mitochondrial dysfunction rather than blaming certain genetic mutations, right? Again, again, that's a complete departure from the current dogma of cancer being outcome of random genetic mutations, right? And again, the purpose is to rightly pinpoint towards the right underlying causes of cancer and how we are actually can help even oncologists, bring more accuracy in their treatment by combining with nutritional therapeutics, right? So nutritional therapeutics should always precede the standard of care treatments, right? And again, that's a different discussion altogether. But let us focus on what we are right now, uh, come here to discuss, right, about biological age and longevity. Yeah.
Yeah, so folks, today we will be talking about biological age, and this discussion will encompass that how we have built the biological age model, what kind of data sets we have taken, right? But before that, Sushant, let us understand very basics in a layman's language. What exactly is aging, and how are we measuring longevity in India? How are we measuring the parameters of biological age? Because as we have seen, there are a lot of tests, blood tests, and applications, and a lot of other things that are there as of now in the market, right? So we
Mm-hmm.
would like to know what exactly is the current scenario and how accuracy and precision are these kind of tests and applications providing to the customers, right? And why should a person be tracking their biological age? Why is it so much of significance? Apart, as long as you're a normal person living under normal circumstances, doing a job, and like a householder, right? Why would you want to go for something called biological age, right? You're not from the entertainment industry, you're not concerned about how you look, right? For all the normal people, but is it related anywhere to the health and disease aspect for human body? So please enlighten us here.
Okay, so first of all, let us uh, define what aging is and how it is linked to health. So, in simple layman terms, aging is a progressive decline in the way our cell functions, right? So, understand the unit of life is cells. Now we have 38 trillion cells, right, inside our body, and more than 50% of the cells, around 57% of cells are microbial, right? So every day whatever we do is impacting our cells, right? And our body are tuned to regulate those external manifestations and external provocative insults which we do every day. Unknowingly we are doing it, right? Obviously not knowingly, because we won't harm ourselves, right? Knowingly. So it is an time phase deterioration of cellular functions, a progressive deterioration of cellular functions, which is inevitable because we, as we pass every single day, our cells are aging, right? That's a normal process. But why we focus on aging right now too much is that we are actually aging faster than what we should, right? So probably there is a chronological age which is defined by our birth date, the day we are born, right? And we every year we grew older, right? But biologically our cells have a different story to tell, right? So they may be, in simple words, for people who do not understand too much of biology or do not want to understand simple words, you know uh, based on your everyday lifestyle choices, you are degrading your cells, you are stopping your cells from performing essential functions, you are interfering with a lot of signaling aspects and the biochemical reactions, which is degrading your cells. And again, this is what is making you age faster than your chronological age, right? So mathematically, your biological age should be less than your chronological age. If you live longer, that is a normal definition of longevity. And this again is an outcome of millions of transactions, biochemical transactions happening inside the body, right? So coming on to health, aging is yet another disease. It's a disease, right? It is so there is a deep rooted correlation between your health and aging. So do not think that if I am going to gym, I'm doing six pack, I have six pack abs, or I'm in entertainment industry, or I'm in a model, or probably I want to look great, I want to feel great, and I will resort to certain fad interventions, or the unscientific claims, right? It doesn't mean that you are not going to age, or it doesn't mean that these aging concept only resort to the people in fitness industry or people in the entertainment industry, right? It is a lifestyle disease, right? So some of the age related diseases are cardiovascular diseases, is the age related disease, right? Which we now have seen over the last couple of years that this disease is being preponing in at least 5, 10, 15 years, right? So we are developing, so people in the age of 25, 30, 35 are developing
40s, like
cardio.
40s is a very early age for something like cardiovascular disease. And we have seen that, people who nowhere have experienced symptoms of cardiovascular early during their life, suddenly one day they are suffering from heart attack. There have been so many incidences in weddings and in events where, out of nowhere they've just experiencing a heart attack, and, it has led to deaths.
Mm.
So, I mean, this is really alarming, and I'm pretty sure this is something to do with the cellular functions of the body. On a molecular level, we are already tracking, right? At Gene Fithletics, we are already providing the insights at molecular level around various pathways that impact the body. But on a cellular level, what kind of insights can a person, avail when they are able to understand the significance of biological age?
So I'll just give a trailer to it, and then probably we can first get into the gaps or the current assessment procedures, and where the gap lies and how you're filling this gap, right? So ideally, what happens is that we know that our cells have nucleus have DNA, which is organized into genes, right? Our genes is static, doesn't change, but there are external factors that may lead to underexpression or expression of genes. So coming into basic here, and I'm just going to few 75 years back, right? There was a researcher called Ansel Keys in 1958, right, who said that cholesterol causes heart disease, right? And he did a survey on 22 countries, and he found that out of that 22 countries, 7 countries had a high rate of mortality of cardiovascular disease, and that was attributed to fat, right? But what he didn't, opened up or shared that fat was trans fat, right? Hydrogenated fat and bad fats, and and then he had hit that it is only, I did research only of 7 countries, and 7 countries had all this, and then that became a standard of care, and all the healthcare practitioners followed that. Cholesterol causes heart disease, and the focus has been on the suppressing cholesterol, right? But now, why I'm talking about how it is linked to aging is that your cells, right? Your cells have a cell membrane, right, which is again a jelly type structure, and there we have to maintain the cell membrane structure and fluidity, right? To maintain that, it needs a lot of lipids and cholesterol, which allows for cells inside to sense what is outside, to stimulate intracellular signaling, to activate certain genes, to activate certain proteins, to perform functions, to keep your body in homeostasis, right? So again, by the very fact of resorting to the pharmaceutical interventions to suppress cholesterol, or probably blaming fat as bad, right? Obviously, there are certain fat, depending on the chemical structure of the fat, which can be harmful. But again, blaming everyone is a disaster. What we have done, right? So that is one basic thing. So what you understand, what in simple words, there's something called cellular proteostasis, right? So it means how your cells make, fold, deliver, transport or degrade proteins, right? So here I do not mean by amino acids. Body make proteins to perform functions, right? So again, that is one aspect what we look at, right? We look at the oxidative stress markers, which is causing cellular stress and cellular decay, cellular senescence, right? Again, which is your ability of your cells or your body to repair and replace dysfunctional cells, to replicate and reproduce more cells, right? So there are multiple few things, telomere shortening, which we'll discuss about it also, that a lot of companies do telomere shortening or DNA methylation tests, and but again, these are just few aspects, right? But again, coming back to our discussion of how we do in elaborated way, probably let us first educate on the current practices, and why it is not giving you the current, or probably these are not based on existing any data set. So what we are telling you is based on the existing data sets which we have collected, mined, and processed, analyzed, and interpreted over the last 5 years.
That's correct, actually. So I think we should stick to the conversation where you're trying to fill in the gaps of the current solutions.
Mm. Okay, so let us understand few things here. So for we have handful of companies now launching biological age factors, right? Biological age clock, right? And the most prominent ones are, there are three categories here, right? One is DNA methylation. So what I mean by methylation is the under expression or no expression of certain genes, right? So these are again caused by certain external insults and external provocative factors, lifestyle choices, the way we live, the way we sleep, the way we eat, the people we hang out with, our state of our oral health, number of things that may lead to underexpression of certain genes. That genes could give you good things or bad things, but mostly underexpression is the good expression of the right genes which are required to perform beneficial functions are being underexpressed, right? So again, this is one of the factors, right? So this doesn't give you a comprehensive view of your biology, of your cellular biology, right? Second is the telomere shortening. Again, we look at, there is a cell division takes place, and then telomeres. Every time there's a cell division, the length of telomeres, which is the end of the chromosomes, which is a protein structure at the end of the chromosomes, which required to protect the cell integrity, right? And again, we over period of time, telomere length shortens, and again, this is one of the factors which people look at to measure biological age. Again, it is not; it is just one of the factors, right? Because cell degradation or the progressive decline of cellular functions, they are this is one of the factors. There are multiple factors, right? So even if you're measuring telomere shortening, it doesn't tell uh, how bad your mitochondria is doing. So again, mitochondria, if a lot of people know mitochondria is the energy powerhouse. It is that particular organelle, ancient organelle, right? Which is one of the bacterias which sits in cytoplasm of the cell, and it actually translates the energy sources into adenine triphosphate ATP, which is an energy for our cells through which we live. So energy, period. Energy is everything. Without energy, you cannot survive, right? So mitochondria is the center powerhouse, or the powerhouse of your or battery of your cells. It gives energy. Again, so it the telomere length, measuring telomere doesn't translate into something. So the idea is not only to measure biological age, but again, if it's not going well, if it's exceeding my chronological age, what specific interventions I need to address it. And until unless you do not measure your biology at a system biology level, at a systemic view, right, you can't address it. Third thing is again, I laugh at it. Is a lot of people are looking at blood works, right? Again,
You
you know,
actually elaborate here what kind of blood works? Is it the normal lipid profiles are we talking about, or is there a
yeah,
blood
yeah.
work?
So the people look at HbA1c, people look at HDL levels, triglyceride levels. People look at these markers, and again they may look at your wearable stuff, right? But the point here is that none of these factors measure. So understand that we get 25,000 genes from our parents, right? When we are born, we get 20 million genes from our microbiome, right? So none of these markers actually measure the specific impact of the microbiome or the impact of oxidative stress or reactive oxidative species, right? So idea is you are using them, but these are not telling you specific reasons for decline in your cellular functions, or how do you derive any solutions based on any biological, any bio and these plasma level tests, which itself does not measure your microbiome. And because these people who are doing it, they still do not accept the fact that microbiome drives our bodily functions, right? And another important aspect here is you have to understand and that's why I always say that the blood test or the plasma or the standard diagnostic test what we currently have should not be the starting point, should be the ending point. And the reason is obviously you start with a particular how a particular action impact your cells, which leads to expression of protein, which lead to biochemical functions, which can stimulate your immune system, your mitochondria, which could lead to certain production, certain molecule which may enter your bloodstream, and that is what they measure, right? So until you know what specific biochemical function is causing that particular inflammatory molecule or inflammatory cascade, it is of no use. Second thing, you have to understand, how a particular diagnostic test takes place, right? So the way the sample is taken, stored, transported, shaken, processed, there are more than 140 steps, right? And each of these steps have a bearing on your test results, right? So if even one steps goes haywire, or doesn't meet the required protocol, your results are going to be away off the hook, right? And again, it is so much so that there is a standard deviation of 30%, right? So if you go for a same test at two different labs, there will be two different results. Now, which to believe, which not to believe, which is accurate, which is not accurate, you do not know, because even they are marketed as enable accreditation, but again, enable accreditation is for the process, not for the results, right? So there's no guarantee. And again, based on if you are doing biological age based on these markers, which does not take care of your cellular functions, do not take care of your, do not measure your microbiome functions, do not measure your mitochondria, and the result itself are distorted, how can you even measure a biological age based on these blood markers, right? Plus there is no data sets on Indian population which is actually using this to measure biological age. It is just a way to show up in this market because you have this fear of missing out, right? And it is all because there's a longevity wave. So this
I
is
understand. I can totally relate to what exactly you're trying to say here as a layman. And
Mhm.
also it's very there has to be this awareness about the fact that lot of the blood works, there might be false positives, there might be false negatives. I mean, sometimes a person who is anticipating that, I have so and so problem might end up having results which are perfectly fall within the normal range, but it could be a false negative.
So this good thing which you brought. So when we talk of range, so
Yes.
it may take a normal abnormal range. What do you mean by range here? It is not optimal. It is comparing your results with other unhealthy population. So whether you are less worse or more worse than those unhealthy people, it is what is telling. It is not telling you this is optimal, right? Because the industry has never focused on identifying the optimality of a particular test, right? So for instance, if someone has a HbA1c of say 5.9 or 6.1, right? They may be traveling for last 3 months, and they may have certain hyperglycemic reactions because of your dietary choices or because of less sleep or whatever it is, right? But again, these cannot translate into a diabetic situation as such, right? Because these are based on last 3 or last 6 months, right? Again, so there is no optimality. So that's why, see, leave biological age. These cannot even tell you whether you have a heart disease or type 2 diabetes or chronic kidney disease or whatever it is, right? So again, see, if you're not measuring your cellular functions, if you're not measuring your microbiome functions, you cannot tell your biochemical age, your biological age, or your biological age clock. So it's all, and there's no data on it. So please, if you have, probably show the data, So again, that is something, what I want to convey is that there are certain biological clocks which look at good things, but not a comprehensive view, right? And there are certain which is generally absurd, which is just a way to show up in the
So
market.
what we have understood here is that I mean, people are working in bits and pieces. It's mostly in the isolation, right?
And there's no data. So the point is, where is the data? So a lot of these DNA methylation tests is a copied model from the West, right? So there's no, where is the Indian
Yeah,
specific
assuming
data on
that
that?
the kind of data that they are taking is that data from the West. It's not the native Indian data, let's say. I mean, that's what is the kind of understanding that one can have from these tests. So,
Hmm.
Sushant, I think we've covered this aspect of where we are filling the gaps. Now, let us educate the audience about how we are measuring the data sets, what kind of data sets we've taken, right? Because this
Okay,
is
okay.
fully based on Indian data. So, folks, our newly launched aspect for biological age is called ageIt. So, it's actually a feature which is provided alongside our mathematical algorithm called Protiva. And this is actually a machine learning model. So, it keeps on revamping every time
So,
we have a.
so I'll hold you here. So people will think machine learning is a myth, and people just talk about machine learning. I'm going to open up to you two things here,
Sure.
right? I'm going to tell you what mathematical models we have used. We have used here single value decomposition method and uh, square matrix, vector matrix methods to mine data, to draw insightful comparisons, and to draw the specific, quantify the specific ranges, right, based on Indian population, which drives the contribution towards how we are aging at cellular level, right? So from a machine learning perspective, that is there. So again, uh, from your question perspective, we are going to here focus on four major aspects. One is the vital cellular functions, right? The markers which we measure to assess vital cellular functions, right? Second would be the inflammation, the systemic inflammation, which can be from a gut microbiome, oral microbiome, or the external chemicals, or the polluted environment, right? The pollution part, right? Or the packaged food which are filled with a lot of chemicals, or even the hydrogenated oils, or the omega-6 oils and the seed oils which have inbuilt chemicals, which is impacting how your uh, cells are being impacted. The oxidative stress markers, so there is oxidative stress. It could be due to an excess exercise, or it could be due to a certain uh, fat diets, or certain ingestion of certain compound which is causing oxidative stress, which is producing certain metabolites which is causing oxidative stress, right? And then also the rate of the production of antioxidants inside your body, which is again your gut microbiome comes into picture, how it is less than the rate of the production of reactive oxygen species, right? And then we also look at cellular oral microbiome markers, the cellular stress, where I'm going to talk about cellular stress. So we'll look at the unfolded protein response. We'll look at the hypoxia-induced stress, the genotoxic stress, the metabolic stress, number of these stress factors, right? So let us start with basics here. So first thing is what as I mentioned, was cellular proteostasis. So obviously your cells have a capability to make, fold, you know, transport, deliver, and degrade protein. So excess protein or misfolded proteins can be degraded. That is a normal process, right? And again, so we were discussing about how do we start with these cellular pathways, right? So we look at the specific external factor which is leading to epigenetic variation, which is lighting up certain pathways, and those pathways is what impacts our cellular functions. So first thing what we look at is cellular proteostasis. So again, looking at how your cells, ability of your cells to make, fold, transport, deliver at different places, or degrade certain proteins, maybe extra proteins or misfolded proteins, right? Uh, so accumulation of these misfolded protein is what we call is you know unfolded protein response, which is again UPR, which is again cellular stress response, and again it is caused by certain. I'll not name all of them, but certain pathways we look at is the production of precresol, which is microbial byproduct of tyrosine. Indoxyl sulfate is the indole derivative, which is a microbial byproduct of tryptophan, right? We look at other cellular senescence pathways like phenylacetyl-glutamine, which is again a byproduct of microbes, as fermenting phenylalanine, which is again an essential amino acids, right? We look at DNA stress marker, oxidative stress marker. So we do look at the production of TMAO, the lipopolysaccharides, right? Pyrrhotyrine, so protein fermentation levels, right? We look at pyrrhotyrine, we look at ammonia, uh, sulfide production pathways, right? Even these could lead to a lot of oxidative stress. So we look at oxidative stress markers, right? So you see, these have talk, together in a very, very, coordinated way. So looking at the misfolded protein response, the unable of cells being unable to degrade the misfolded protein or the extra protein, right? And again, these are normal process where the body regulates cellular functions, and through mitochondrial health, promoting mitochondrial health through various cellular and molecular intervention, and nutritional interventions and molecular interventions where they could uh, boost mitochondrial biogenesis. Again, I'm going to talk about as how we stimulate mitochondrial biogenesis, activating another gene, which is PGC1 alpha, which is a hallmark for longevity, right? uh, Allowing cells to repair and replace dysfunctional cells, allowing cell to replicate and produce more cells, right? And then autophagy, number of these things, these functions which we measure the ability of cells or the external or the certain molecular pathway which is hampering this ability of cells to perform these functions, right? So we look at various these pathways where we look at for instance, homocysteine levels, right, which could lead to overexpression of asymmetric dimethylarginase ADMA, which suppresses nitric oxide production, which is again impact your role on longevity, right? So DNA damage, oxidative stress markers we look at, right? Then coming on to telomere shortening, stem cell regenerative signaling, hypoxia stress. So I'm going to discuss briefly all of these, right? So let us start with, I will explain these cellular functions, these inflammation pathways which could impact your cellular functions, right? Repairing cells, right? For instance, stem cells, mobilization, differentiation of stem cells into different tissues to replace and repair dysfunction cells is actually your nitric oxide produced either alongside your lining of blood vessels, right, or produced by oral microbiome via nitrate reductase pathway, right? It could actually nitric oxide stimulates or the stem cell mobilization differentiation, which helps in repair work, right? Similarly, we have this nitric oxide also produces another secondary messenger called cyclic GMP, which activates PGC1 alpha, which is actually leads to mitochondrial biogenesis. Nitric oxide, your nitric oxide actually, promotes or controls electron transport chain, right? And prevents electron leakage. So it also improves mitochondrial efficiency, right? So again, then telomere shortening, nitric oxide prevents telomere shortening, right? So it activates telomerase, which is an enzyme which inhibits telomere shortening, right? So again, uh, uh, you know, uh, we measure the actual nitric oxide produced, you know, by oral microbiome, right? And also see the inflammatory factor, which is causing endothelial cell dysfunction, which is causing the uh, functional loss of nitric oxide via the ENOS nitric oxide synthase pathway, right? So this is we can measure the telomere length, we can measure the specific senescence factors, which is impacting telomere length. We can tell you your stem cell regeneration capacity. We can tell the oxidative stress markers, which is causing DNA damage, right? Regenerative signaling, right, where cell repair and cellular dysfunction or cell replacement happens, or maybe cell replication happens, right? So we measure all these pathways which is impacting these functions, right? And again, antioxidant production, what we look at. So we generally look at your gut microbiome converting polyphenols, flavonols, right? Catechins, a lot of these substances into antioxidants, which can suppress your oxidative stress, right? And again, so these are few of the factors we look at, right? Where we measure the inflammatory and oxidative stress markers, which directly impact your cellular pathways, right? And protein fermentation levels we look at, right? In terms of whether your microbiome is actually converting lysine into cadaverine, right? Or producing putrescine or ammonia, right? Or maybe there's a lot of hydrogen sulfide production either by oral gut microbiome, which is again can lead to apoptosis. Or another thing which I forgot to mention was, right, so your nitric oxide is actually delivers oxygen and nutrients to various cells and tissues and organs, right? So a lack of or functional loss of nitric oxide could lead to hypoxia related stress, which could lead to tissue death, right? And again, so cellular stress, as I mentioned, unfolded protein response will look at various pathways which is causing unfolded protein response, right? Cellular senescence pathways. So we all are measuring at cellular level, right? And we combine them statistically together to arrive at, again, the machine learning model which we have used, right? Whether using single value decomposition method, is we arrive at a particular number, which is again based on the our internal validation study we have done on 6 billion molecular data sets, right? Which help us measure these pathways. And again, we do not stop there. So we measure each of these pathways and then modulate. So in terms of regulate them or stimulate the expression of certain pathways or suppress the expression of certain pathways based on certain molecular and nutritional ingredients. Right. For instance, if you have a high protein fermentation level, it could involve, you know, glycine betaine, which actually, trimethylglycine TMG, which actually can suppress your protein fermentation level. But again, if you have a lot of high methane gas production, probably this will not be usable. So we can look at uh, other nutrition substances like berberine, or probably for promoting mitochondrial genesis, you can look at not only NAD, which is again a supplement, right? Again, which may or may not have an impact on your mitochondrial biogenesis. Depending if there's a lot of inflammation, cellular senescence, probably NAD will not work, right? So we can look at ursolic acid, right? Or maybe other specific flavonols, or the flavonoids. We can look at things in cocoa which can stimulate mitochondria, which can stimulate nitric oxide production. It's a hell a lot of biochemical signaling what we look at inside the body at a molecular and cellular level, right? And again, and so that's why we provide a very comprehensive view where we look at your oral microbiome functions, your gut microbiome functions, your mitochondria, your stress level, whether it's hypoxia stress or cellular stress or genotoxic stress or metabolic stress, right? So again, this is how we are filling up this gap.
That was pretty interesting and scientific terminologies that you've used here. I'm sure that most of the people from the industry could relate to it. And yes, of course, because it's very important these days to understand that what exactly is the kind of pathway impacting the biological age, so that one can make an informed decision, right? And what is more fascinating is that when we are giving this data in the front end for a customer or a user, so we show them that these are your pathways which are impacting your biological age.
So
Yeah.
again, it's so I'll just clarify here. So one pathway doesn't mean one. So one pathway may have thousands of biochemical. It will be culmination of or constellation of 5,000, 10,000 biochemical functions. So every microbe would have on an average 2 to 3,000 genes. Every gene would be underexpressed, overexpressed, or not expressed. So there may be a pathway may have more than 1,000 genes, 1,000 bacteria, right? Or 500 bacteria, or 50 or 100 bacteria. Each bacteria may be having maybe 2,000, 3,000 genes. Each genes, or maybe out of that 1,000 genes, may be underexpressed. Some may be overexpressed. So we look at, so one pathway would be culmination of 10, at least 10,000 biochemical functions, right? So so again, it is not measuring one pathway. And when we, as Sakshi, as you rightly said, right? So uh, we show, because there's so much data, we show only top few pathways which is actually impacting the
And
value
for
chain.
every individual, it will vary. I mean, as we are building the model already, it's being fed with every time there's a new data in the back end, time there are new data sets, new molecular pathways coming to us. It's constantly, these pathways will change, right? Their efficiency will change, their effectiveness will change based on whatever new research is coming around, all of these biochemical functions. So, I mean, I think in true sense, this is truly a very practical model that we have built here for the benefit of the users. So, Sushant, I think we've explained it all. We have also told that what kind of therapeutic interventions one might expect. So, just for the sake of clarity, for all the users, it is actually a nutrition, it's a combination of the nutrition and the supplement recommendations that we recommend, right? So Sushant, you'd like to add here something?
Yeah, as people who have signed up for our product or who know, who follow us, so what we do is it is very unsupervised. There is no human intervention here, right? So based on the molecular data, the phenotype metadata, the clinical data which we collect, right? We combine them, and then we convert. We Our recommendation in engine marries these pathways activity expression level, which is, there are maybe more than 500 plus pathways which we look at, right? Some are shown on front end, some we do not show right now because of certain patent pending right now, right? So these are our recommendation engine maps these with the nutrition ontology, which is again a ontology of 40,000 plus molecules, right? These molecules are those which are found in natural food we consume, whether it is vegetables, whether it is lentils, whether it is fermented goods, beverages, protein sources, fat sources, right? Seeds, herbs, right? Then specific supplements. So when we talk of, so what we do is we marry them, and then, for instance, as I mentioned, for glycine betaine, so probably if you have high protein fermentation level which we measure, right? So we could recommend the foods or supplement in this. Again, we also look at methane. So probably if your methane methanogen activity or methane producing bacteria activity is very high, probably we may not recommend it, right? So essentially, similarly, we may recommend NAD, or probably we may not, based on, if there are certain oxidative stress and inflammatory markers are overexpressed, right? Or probably uric acid is high because NAD has niacin, so niacin is harmful for you, for people who have high uric acid levels. We do measure uric acid levels as well, right? We look at your homocysteine levels to actually to tell, and also look at your vitamin B6, B12 absorption to tell whether what is causing the high homocysteine levels, right? And these homocysteine levels actually were one of the biomarker for cellular senescence, right? And again, it causes an age-related cardiovascular issue, right? Because it suppresses nitric oxide production, right? We In terms of supplement, what we recommend, so we recommend specific amino acids, adaptogens, minerals, vitamins, food extracts, prebiotics, probiotics in specific quantity, in specific frequency. And again, here probiotics we recommend strains, right? We do not recommend, like other company species, there are specific strains based on your pathways, which is map your pathways, and again specific quantity. So these are not standard sort of supplements which we recommend that most of other companies are selling, right? So again, these are meant to modulate certain pathway which is going to suppress inflammation and oxidative, which is going to promote mitochondrial biogenesis, which is going to stimulate nitric oxide production, which is going to suppress cellular senescence, which is going to actually lead to a suppress hypoxia induced stress or other metabolic induced stress or genotoxicity induced stress, right, or cellular stress response, right? So these are meant to impact various more than 500 plus pathways, which is going to actually impact your biological age, right? And then we every time you retest, or every 6 months you retest, or it is your choice whether you want to test in 6 months or 12 months. Then probably we recalculate your biological age, right?
That
Based on
I think that's we have given a complete overview of how the platform works and what exactly are the kind of insights one can get. So thank you, Sushant. That was a pretty insightful discussion. It was a very detailed and very comprehensive discussion on the biological age aspect of what we are offering. And also for all our listeners, thank you for listening to our podcast. We are you have shown us love on YouTube, Spotify, Apple Podcast, and thank you so much for that. And Sushant, would you like any last words would you like to add before we conclude?
Yeah, so thing is there are a lot of now last 3 4 years a lot of things coming up with respect to health, a lot of supplements, and now tests, the biological age applications coming up. But just ask the right questions, whether you have the right data sets, where the science at least ask for the existing scientific literature which is backing their claims, right? I understand there may not be possibility of everyone having an India specific data sets, right? Because we also took 5 years to build that, right? So again, uh, at least you should have certain scientific relevance to it, right? Because there are a lot of obnoxious claims, unscientific claims, wrong information spread across the entire spectrum of healthcare, right? And again, we need to ask the right questions. For instance, this we have discussed a lot of time. When it comes to protein, we are thinking we are protein deficient nation. All these things are coming up. You have to understand, it is not about consumption. It is about absorption, right? So again, you are knowingly and knowing consuming a lot of amino acids, right? You have a lot of sources. Thing is, if you are not getting benefit of it, it is because of certain issues in your biochemistry, within your microbiome, within your lack of certain digestive enzymes, right? Or there's high protein fermentation level, or certain bacteria which is transforming your amino acids, fermenting into harmful toxic metabolites, right? So again, also another last thing I want to add here is that in terms of these smart wearables, what we have, right? And now the trend is moving from watches to rings, right? And again, there are rings which are tracking your HRV levels, your blood pressure levels, your sleep levels. The question you should ask, and as a layman, I'm telling you, if I have to buy this so I can measure my sleep, let's assume it is deficient, so what interventions I have to regulate it? Because these variables will not tell you the source of this dysregulation, right? For instance, if I have a lack of sleep, is it caused by production of certain neurotoxins by your gut microbiome, or is it consumption of fluoridated toothpaste which is causing disrupting melatonin production, or it is low GABA production, whatever it is, right? So there are multiple reasons. Similarly for HRV also, right? What is causing is fluctuation in HRV, right? What specific factor is it? Nitric oxide, it is LPS production, it is GABA production, number of these things which can impact, right? So again, blood pressure, again, for blood pressure and hypertension, because this is impacted to a lot of people, right? Two things before we sign off, right? I want to give a few liner here. Just think over it for all those who are listening. Hypertension and vitamin D absorption. Hypertension is impacting at least 91% of the Indian population. And what we do is we try to remove salt from our diet, or we try to track our blood pressure. The problem is oral health. The problem is your toothpaste. You are using toothpaste which are high in fluoride, which is high in saccharides, aspartame, SLS, a lot of these harmful chemicals. which is disrupting your nitric oxide production, right? Which is so nitric oxide it dilates blood vessels. So if you do not have nitric oxide production, it is going to lead to vasoconstriction, and it is going to increase your blood pressure and hypertension issues. So fix your oral health rather than just tracking it. You know that. So try to see what is happening in your whether you have a dry mouth or not. You know, dry mouth can impact your blood pressure levels, right? Do not stop doing mouth breathing, right? That is again another problem. Coming on to vitamin D, right? Vitamin D, there are a lot of capsules, a lot of supplements, and a lot of sources of both animal and plant-based sources. But again, it is not the active form of vitamin D. Active form of vitamin D is something your bitrate producing bacteria converts vitamin inactive form into active form of vitamin D. So a lot of people who have vitamin D issues are because your gut microbiome is imbalanced. So there are certain gut bacteria which produce bitrate, and which is also stimulates or also biotransforms inactive to active form of vitamin D. Right. So this is something which you folks should understand. You know, this is what I want to say. Probably we do measure vitamin D levels. We do measure vitamin D absorption levels, not vitamin D consumption. We can tell you whether even if you're consuming vitamin D related foods, what is the absorption level, whether you have the right bacteria doing right functions, expressing right genes to actually uh, give you active vitamin D or not. Yeah.
So thank you. That's all for today. We'll see you folks in another episode. Thank you, Sushant.
Thank you.